Oh yeah, now it's recording. So today we'll be comparing and contrasting the structures of bacterial and eukaryotic mRNAs and how they're transcribed.
So first we're going to go over some basic mRNA information. And according to the central dogma, the transition from DNA to RNA is transcription, and then from RNA to protein is translation. And mRNA is produced in the transcription process, and it's basically a single strand of RNA molecule that codes for protein.
Also, eukaryotic mRNAs are monocistronic, which means that one gene can only code for one specific protein. Meanwhile, bacterial mRNAs are polycistronic, meaning that one gene can code for multiple proteins.
And then another thing to note is that eukaryotic mRNA is transcribed in the nucleus and then transported to the cytoplasm, whereas bacterial mRNA is transcribed directly in the cytoplasm.
So now we'll be going over the process. So for eukaryotic mRNAs, it begins with the 5' capping, which serves to protect the mRNA strand. And after, transcription factors help the RNA polymerase enzyme recognize the promoter, which has the TATA sequence. And what the RNA polymerase does is that it unwinds the DNA strand for the next step.
And then next is the encoding process. So you can see here that basically the nucleotide bases are matched to their respective counterparts on the mRNA strand to the ones on the DNA strand.
So after it's done being encoding, the mRNA strand actually has a lot of introns, which are the non-coding excess regions of the mRNA strand. And what RNA splicing does is that it removes these introns and stitches the exons together, which are the coding regions.
And then after splicing is the poly-A tail process, and this basically modifies the 3' end of the mRNA strand. So first an enzyme comes in and trims it, and then a second enzyme comes in and adds the adenine nucleotide bases that you can see here, which is the actual poly-A tail part of it. And this process basically helps with structural stability of the mRNA strand, and then it also prepares it for transportation to the cytoplasm.
So for the bacterial process, it does not require the 5' capping to start, but it does require transcription factors. And for the bacterial mRNA, these are called sigma factors. And it does the same job as the other transcription factors, which allows the RNA polymerase to recognize the promoter. And again, just like eukaryotic mRNA process, RNA polymerase opens up the helix.
And also the mRNA transcription process is a lot simpler in bacterial cells than it is in eukaryotic cells. So basically, bacterial mRNA does not contain introns, so there's nothing that needs to be removed, and they skip the splicing step altogether.
And because they don't have any introns, they skip the splicing step, the bacterial mRNA is ready to be translated in the cytoplasm. And that's it.
Nice.
Yeah.